Good afternoon, everyone. It's a great honor for me to have this occasion to present our work on behalf of Professor Chunhua Zhao and our team, focusing on the application of post-embryonic pluripotent stem cells for the treatment of major diseases, including those related to aging. As other speakers have highlighted, in treating complex diseases, we have entered the era of cellular medicine. Among all cell-based products, stem cells are particularly valuable due to their potential for tissue repair and regeneration, immune modulation, inflammation reduction, and oxidative stress mitigation. These are actually the key mechanisms that make them promising candidates that for anti-aging and other therapeutic applications. And this has driven a global surge in stem cell development over the past decade. In the early stages of developing stem cell technologies and therapies, we faced major challenges, from sourcing quality stem cells to advancing key engineering techniques and ensuring the safety and efficacy of stem cell products. For the first time in the world, our group leader, Professor Zhao, leading our team, identified a subpopulation of stem cells that exist after the embryonic stage of development. We term them as post-embryonic pluripotent stem cells, PPSC. Since PPSC have the capacity of differentiating into all cell types within the ectoderm, mesoderm, and endoderm, we propose they are the ideal seed cells for cellular medicine, engineering, and industrialization. We characterized the biological properties of PPSC at both genetic and epigenetic levels. Key phenotypes, including positive expression of CD29 and negative lean expression, support their stemness and multilineage potential. Importantly, PPSC lack expression of OCT4, have weak NAMOC expression, and exhibit high KLF4 levels. This gene profile mitigates tumorigenic risk while retaining sufficient stemness, supporting both the safety and differentiation capacity of these cells. Upon further exploration, we discovered that PPSC exert both anti-inflammatory and pro-inflammatory effects. For instance, PPSC can inhibit the differentiation and maturation of dendritic cells, while also promoting their function by inducing the generation of a subpopulation of regulatory dendritic cells. This important characteristic enables PPSC to maintain tissue and cell homeostasis. So we propose that these bidirectional immunoregulatory effects serve as a theoretical basis for the effectiveness of PPSC in treating immune disorders. These findings are listed in the faculty 1000 database and have received widespread acknowledgement and positive feedback from internationally recognized specialists and institutions. In the special reports in immunity, the discovery of PPSC was highlighted as offering promising therapies for major diseases, including organ rejection and autoimmune disorders. Additionally, our research results have garnered worldwide attention and have been cited in numerous peer-reviewed publications by esteemed figures. For the industrialization of PPSC, our R&D team established China's first industry standard and the core system for the large-scale application of adult stem cells, accompanied by a series of patented technology standards. Due to our identification of PPSC and the corresponding drug production standards, we have received numerous awards and have been recognized as an industry leader in adult stem cell engineering. In 2004, we obtained China's first approval for stem cell clinical trial aimed at treating leukemia, followed by another approval in 2006. Our long-term clinical studies demonstrated the safety and stability of PPSC, with the overall treatment efficacy for leukemia reaching up to 88.9%. PPSC-derived drugs have also demonstrated significant potential in treating severe cases of graft versus host disease, GVHD, particularly those resistant to traditional hormone therapies. Due to these positive effects, we received the first approval for a phase 3 clinical trial of stem cell drugs from the FDA in China. Over the past 16 years, these trials, conducted at multiple national hospitals, have confirmed the safety and effectiveness of our stem cell drugs in treating GVHD, achieving a treatment efficacy of 78%. As our indications expand, all PPSC-based pharmaceuticals undergo rigorous and repeated evaluation for clinical effectiveness. For example, the stem cell drug for blood disorders has been clinically validated for 15 years, while the drug for heart diseases has successfully treated over 100 cases. Long-term efficacy and standardized research protocols remain our primary evaluation criteria. Based on our PPSC technology and promising clinical results, we successfully developed China's first, second, and several other stem cell drugs targeting severe and refractory diseases. These drugs have been shown to be effective in preclinical studies and are currently undergoing extensive evaluations across all stages of clinical trials. In response to the global public health pandemic in 2019, particularly considering its disproportionate impact on aging populations, we have been leading key projects to investigate the potential of using PPSC to reduce the severity of COVID-19 and its related complications. In a pilot clinical study, we showed that MSC, mesenchymal stem cells, prepared through our PPSC system significantly alleviated COVID-19 symptoms, reduced hospital stay durations, and increased cumulative remission rates. The therapeutic mechanism involves the suppression of overactivated immune cells and regulation of inflammatory cytokine release. Importantly, PPSC-based MSCs exhibit minimal ACE2 and TMPRSS2 expression, reducing their susceptibility to coronavirus infection, and their high expression of multiple growth factors underscored their strong immunoregulatory capabilities. Following our pilot study, we conducted a randomized, single-blind, placebo-controlled phase 2 clinical trial to further assess the safety and efficacy of ACE2 negative MSCs in treating patients with severe COVID-19. In the peripheral blood of patients treated with MSC infusion, we identified a unique group of cells expressing VNN2, a marker associated with immune and hematopoietic processes. This finding suggested the mobilization of hematopoietic stem progenitor cells from the bone marrow in response to MSC treatment. Further analysis revealed that this key subpopulation of HSPS, PC, expressed 171 genes linked to angiopoiesis, HSC mobilization, and fetal extramedullary hematopoiesis. Such extramedullary hematopoiesis may support immune function activation, suggesting that this bone marrow-derived cell population could play a crucial role in sustaining immune responses against coronavirus infection. Further supporting this finding, we find PPSC-based MSC helped restore immune homeostasis and promote recovery in COVID-19 patients by modulating various immune cells. They do so through upregulation of chemotaxis-reduced genes in different cell populations, promoting T cell differentiation into helper cells, inhibiting overactivated immune cells, activating B cells to support tissue repair, and suppressing neutrophil extracellular trap formation to reduce the risk of immunothrombosis. To summarize, the molecular mechanism of PPSC-based drugs in improving COVID-19 prognosis involves multiple actions, including combating viral infection, promoting tissue repair, and protecting the body against severe complications such as thrombosis. Our reports on stem cell treatments for COVID-19 were widely recognized by the international community and hold significant clinical application value. Notably, our phase 2 clinical trial was listed as the first reference in the US NIH guidelines for COVID-19 treatment. As a testament to our efforts in utilizing PPSC theory and the system to treat major diseases, Professor Zhao has been recognized as one of Elsevier's most cited scholars in China for 10 consecutive years and was named among the top 2% of the world's top scientists in both 2022 and 2023. Now let's take a moment to review our work on stem cell development and therapies. To date, we have validated the hypothesis and system of PPSC, identified an optimal stem cell seed with immunomodulatory functions, achieved large-scale production of adult stem cells with strong tissue repair capabilities, and established evaluation protocols for clinical safety and feasibility. We feel that our research reflects a commitment to integrity, from the development of theory to the advancement of technology and products, and from those products to industrialization and clinical application. We are grateful for the recognition and support we have received. So finally, I would like to express our heartfelt gratitude to the funding agencies for their support. Thanks for your attention.
Questions? Comments? I will say one thing, and that's thanks to Ilya. During COVID, you do connect me with Professor Zhao, and that's eventually, we can actually confirm your finding. We did a parallel 80-patient study in the US for ARDS COVID patients, and Professor Zhao's initial findings were led also to these studies, showing a significant increase in survival using these cells. So thank you for making all this contact. It's not about aging, it's more than that. Any more comments? Thank you very much.